There are a number of laboratory techniques that can be used to measure the susceptibility of bacteria to antimicrobial agents. Laboratories must use a standardized, well-defined method for performing antimicrobial susceptibility testing to allow veterinarians to select the appropriate antibiotic for treatment purposes. Additionally, standardization and harmonization of antimicrobial susceptibility tests used in epidemiological surveillance of antimicrobial resistance are critical if data are to be compared among national, regional, or international surveillance and monitoring programs. This video provides veterinary laboratories with standardized antimicrobial susceptibility testing methods for bacteria isolated from animals. There are several types of laboratory techniques that can be used to measure the susceptibility of bacteria to antimicrobial agents. These include disc diffusion as well as broth and agar dilution techniques. The common steps for antimicrobial susceptibility tests are preparing chemicals and materials, antimicrobial susceptibility testing, and finally, reading and interpretation of the results. Mueller-Hinton medium is suggested for routine susceptibility tests. Preparation of the agar should be done following the manufacturer's recommendation. After autoclaving, allow the agar to cool down in a water bath set at between 45 to 50 degrees Celsius. For agar plate, pour the freshly prepared and cooled Mueller-Hinton agar, or MHA, into petri dishes to an agar depth of approximately 4 millimeters. Allow the agar medium to cool down to room temperature. The pH of the agar should be 7.2 to 7.4 at room temperature after gelling. Also, check sterility by incubating the first and last poured agar plates at 35 to 37 degrees Celsius overnight. If these plates are not used the same day, store them at 2 to 8 degrees Celsius. These stored plates should be used within 7 days. To prepare direct colony suspension inoculums, streak the bacterial strains on Mueller-Hinton agar to get single isolates and incubate at 35 to 37 degrees Celsius overnight. Pick the top of at least 4 or 5 well-isolated colonies of the same morphological type with a sterile loop. Transfer to a tube containing 2 milliliters of normal saline water and mix. Adjust the density of inoculum in this tube to turbidity equivalent to 0.5 McFarlane, which is equivalent to 1.5 times 10 to the 8th colony-forming unit per milliliter. The inoculum density can be standardized using a densitometer or a barium sulfate turbidity standard. Use the 0.5 McFarland adjusted inoculums within 15 minutes. Growth method inoculum may also be prepared. Please note that the bacterial number is different for different antimicrobial susceptibility test methods. When preparing antimicrobial stock solution, consider the following. Do not use pharmacy stock or other clinical preparation and store it as recommended by the manufacturers. Warm antimicrobial agents to room temperature before opening for prevention of condensation. If possible, the weight should be more than 100 milligrams. Prepare stock solution at concentration at least 1,000 microgram per milliliter or 10 times the highest concentration tested. And store the aliquots of sterile stock at the manufacturer recommended conditions. Consult the latest version of the Vet01 standards for number of concentrations to be tested. Calculate volume, weight, and potency of antibiotics using these formulas. In this assay, drug diffuses out from the disc into the solid culture medium with the selected inoculum forming concentration gradient with highest concentration close to the disc. Disc diffusion test is based on the measurement of a clear inhibition zone. Prepare standardized inoculum of 0.5 McFarland. Dip a sterile swab into the adjusted suspension. Rotate the swab several times and pressing firmly on the inside wall of the tube above the fluid level to remove excess inoculums from the swab. Streak the swab on the dry surface of the MHA. Repeat this streaking 2 more times while rotating the plate approximately 60 degrees between streaks. Let the plate sit 3 to 5 minutes, but no longer than 15 minutes at room temperature. After flame sterilization of forceps, allow forceps to cool before picking up disc. Place the antibiotic disc on the surface of the agar plate using sterile forceps, needle tip, or a dispensing apparatus. Ensure the discs are no closer than 24 millimeters from each other. No more than 6 discs should be placed on a 100 millimeter plate. Invert the plates and place in an incubator within 15 minutes after the discs are applied. After 16 to 18 hours of incubation, measure the diameter of the zones of complete inhibition. Interpret the size of the inhibition zone by referring to the latest CLSI Vet01 standards and report the bacteria species as susceptible, intermediate, or resistant. Agar dilution method is a quantitative measurement of the in vitro activity of antimicrobial agents. Each agar plate contains a different concentration of the antimicrobial agent in doubling dilutions. The result obtained from the technique is termed MIC, or minimum inhibitory concentration. That is, the lowest concentration of antimicrobial agent that completely inhibits colony formation. MIC is usually expressed in micrograms per milliliter. The test media is made from powder. After autoclaving the agar, cool to 45 to 50 degrees Celsius in a water bath before aliquoting and adding antibiotics. Prepare antibiotic solution using a 2-fold dilution series. The antibiotic concentrations in each tube are double the final expected concentration of antibiotic in corresponding plates. For the first dilution, add directly from stock solution, then serially dilute. The antibiotic agar mixture in petri dishes should be 4 millimeters deep. There are 2 methods for antibiotic and agar mixing. Add 2 milliliters of diluted antibiotic and 18 milliliters of MHA separately into a petri dish. Mix thoroughly, ensuring no bubbles are formed. Add 2 milliliters of antibiotic solution into MHA tube. Mix 2 milliliters of antibiotic solution and 18 milliliters of MHA in a tube and pour into a plate, ensuring no bubbles are formed. Leave the agar to solidify at room temperature and keep to 2 to 8 degrees Celsius. Follow the antibiotic manufacturer instructions regarding how many days the plates can be stored. After adjusting to 0.5 McFarlane, dilute the adjusted solution to 10-fold dilution so the concentration of bacterial solution is 1 times 10 to the 7th colony forming unit per milliliter. Distribute diluted solution into a microtiter plate. And apply inoculums to the agar surface by replicator. Make sure that you inoculate the control agar plate first, then start with lowest concentration. After the spots are dry, invert the plates and incubate them at 35 to 37 degrees Celsius for 16 to 20 hours. Ensure that the MIC of the reference stains that their MICs are in the recommended range. record the MIC as the lowest concentration of antimicrobial agent that completely inhibits the visible growth. Clinical breakpoints are used to define isolates as susceptible or resistant to the antimicrobials being tested. Broth dilution is a method in which a suspension of bacterium of a predetermined optimal concentration is tested against varying concentrations of an antimicrobial agent that is usually serial twofold diluted. It aims to determine MIC. The broth dilution method can be performed either in tubes containing a minimum volume of 2 milliliters or in smaller volumes using micro-titer plates. Broth microdilution test involves the use of small volumes of broth is described here. Mueller-Hinton broth with cation adjustments, or CA-MHB, is recommended as the medium for susceptibility testing of commonly isolated bacteria. Prepare MHB as suggested by the manufacturer, autoclave, and chill overnight at 4 degrees Celsius or in an ice bath if it is used the same day. Prepare sterile 10 milligrams per liter magnesium chloride and 10 milligrams per liter calcium chloride stock solution and store at 4 degrees Celsius. Correct MHB's concentration of divalent cation to 20 to 25 milligrams of calcium ion per liter and 10 to 12.5 milligrams of magnesium ion per liter. In general, 450 microliter of calcium ion and 225 microliter of magnesium ion solution is added to 200 milliliters of MHB. Label micro-titer plates from highest concentration to lowest concentration at the top of the column. Add 50 microliters of cation-adjusted MHB with multi-channel pipette into the micro-titer plate except the first column. Fill the first and second column with 50 microliters of double-strength antibiotic solution. Mix suspension thoroughly and transfer 50 microliters of suspension to the next column. Repeat until finished except for control, the last column. After adjusting 0.5 McFarlane, dilute the bacterial suspension by tenfold to get approximately 10 to the 6th colony-forming unit per milliliter. Do 1 to 100 dilution by adding 100 microliter of bacterial suspension into 9.9 milliliter of cation-adjusted MHB to get approximately 10 to the 6th colony-forming unit per milliliter. Optionally, 1 to the 10 dilution can be performed by adding 1 milliliter of bacterial suspension into 9 milliliter of cation-adjusted MHB, and repeat twice to obtain the similar bacterial cell number. Transfer 50 microliter of bacterial suspension into microtiter plate with twofold serially diluted antibiotic solution. When 50 microliter of bacterial suspension is transferred into microtiter plate with 50 microliter diluted antibiotic solution, the final concentration of bacterial suspension will be approximately 5 times 10 to the 5 colony-forming unit per milliliter, or 5 times 10 to the 4 colony-forming unit per well. To prevent drying, seal each tray with parafilm or with a tight-fitting plastic cover before incubation. Incubate the plate at 35 to 37 degrees Celsius for 16 to 20 hours. Remember not to stack microdilution trays more than 4 high. Results should be read after 16 to 20 hours of incubation. First, check that the MICs of the reference stains are in the recommended range, as prescribed in the latest Vet01 standards. Then, record the MIC value, which is the lowest concentration of an antimicrobial agent that inhibits visible growth of the organism in the microdilution wells. Clinical breakpoints as prescribed in the latest Vet01 standards are used to define isolates as susceptible or resistant to the antimicrobials being tested.